Howdy, howdy, Drew Dragons. Welcome to Chapter 4. This is, or should not be, all that difficult a chapter for you. This, I'm hoping, is mostly review, and really what I want you to get out of this chapter is an appreciation for the structure of the cell. What is it made of? What are the organelles? Where are they? And what are their jobs? Okay. There are a few new-ish items that you will see. Those items are probably the connection and communication proteins. I would like you to know them just because later on in the course we are going to definitely spend a lot more time on them. Okay, so let's get started. What do we have here? This is an important concept. This is basically, actually I'm going to stop right here and skip back for a moment. The idea of a difference between a prokaryotic cell and a eukaryotic cell, you should definitely know the difference. That kind of comes right around this beginning part of the chapter. Essentially, prokaryotes, bacteria and such, the archaeans, do not have defined organelles or those complex structures within the cell. They are prior to what we call the endosymbiont theory of cell evolution. That is the currently the most widely accepted theory of how we get eukaryotic cells, which have all those neato organelles in them. That being said, moving on, this is a concept here about surface area and volume. This is why cells can't be huge. Okay, why is it that you don't have a gigantic single-celled organism about the size of an amoeba, which you can barely see with the naked eye, is about as large as you can get, simply because if you look at the math here, let's go for, I don't know, red ink here. If you look at the math, if you increase the cell radius by a factor of 10, look what happens to the volume. The volume goes up by like a factor of 1,000. Okay, it's from 4 to 4,000, and that's because the volume is pi r cubed. Okay, or I'm sorry, 4/3 pi r cubed. But that's a large factor. It's a cubic ratio. So if you have a tiny cell, here's our tiny cell, your volume is going to be, to surface area ratio, is going to be way bigger. Okay, and the problem with that is something that's here in the cell has a lot farther to go to get out of the cell, and something that's coming from here has a lot farther to go to get into the cell. And that's a real problem because cells have got to move molecules in and out all the time, and there's just a functional limit beyond which the cell simply can't move things in and out fast enough. So that's why we're made up of trillions and trillions and trillions of tiny cells rather than several big gigantic cells. Now people might say, well, you have nerve cells. I'll draw a nerve cell looking like this. It's really long, okay? And they can be really long in the legs, for example. I'm like, yes, they can, but the diameter, and I'll click green for this, the diameter around is very narrow, which means that the diffusion rate in and out is still possible. Okay, so that being said, that's why it is that cells are not big. Okay, here is the cell, the typical animal eukaryotic cell. So again, let's go through them one by one. We have the nucleolus in the middle here, and that is primarily going to be the production of a ribosomal RNA for the production of ribosomes. Okay, you have the nucleus, and it should not shock anyone that the nucleus is where the genetic material is encased. It's a protective area. The DNA, the chromosomes, genes, etc., protected in the nucleus. Okay, directly outside the nucleus, you're going to have the rough endoplasmic reticulum. Let's change to red here. So this stuff is the rough endoplasmic reticulum. It goes all the way around the nucleus. It goes around here as well. It's right outside the nucleus because this is the site of protein synthesis. Because of all these little doodads, all right here, these are all these little tiny dots you see on the rough ER, making it look like sandpaper. Those are the ribosomes where protein synthesis happens. That's where the information comes out of the nucleus and proteins are made. Again, more on that later, but that's why it makes sense to make the rough ER right around the nucleus so you have easy access to the information coming out of the nucleus. Next, we have the smooth endoplasmic reticulum. A lot of lipid manufacture and stuff, hormones, etc., happens in the smooth endoplasmic reticulum. Tends to happen farther out. The smooth ER you'll find farther out from the nucleus than the rough ER. Okay, we're not going to go into too much detail right there. The Golgi apparatus. Let's go purple for the Golgi apparatus. Here's our Golgi apparatus. Okay, and oftentimes you'll see it. I'm going to draw it over here. You'll kind of see it. It has shapes like that. Okay, of these folded cristae membranes, and excuse me, cisternae. And these take the protein products that come from the rough ER, package them up, do some modification, and oftentimes will send them for export. Okay, more on that later. Here we have our ever popular mitochondria. Okay, this is where you're going to be producing the cell's ATP. It's the powerhouse of the cell. Change ink colors again. Let's go to orange. We also have the centrioles here, very, very critical in forming the cytoskeleton of the cell and in cell division. Okay, and we're going to have also a lysosome here, the digestive enzyme vesicles of the cell that will carry, again, digestive enzymes and will either break down old organelles or food items that come into the cell. And of course, we'll go for, we never use yellow. Let's use yellow. Of course, we have the cell membrane, which we've already started looking at here, and that is your lipid bilayer membrane. And indeed, all the organelles inside will also be composed of a lipid bilayer membrane. So again, these are the relative positions and functions of the organelles. Excellent. So we have a plant cell, and the plant cell, very similar. The main difference is you're going to have the chloroplasts with the thylakoid membranes inside, or where you have the chlorophyll, where you're going to trap the photons of sunlight and use that energy to make the carbohydrates. You also will have a central vacuole. This is, when it's filled with water, this is what pushes all this stuff out towards the edges and gives plants what we call the turgor pressure that keeps the cells nice and plump and the plant nice and erect, prevents it from drooping. When plants are dehydrated, they droop. If you give them water, this vacuole fills up, blah, blah, blah. Okay, but you're going to see very, mitochondria, Golgi, rough and smooth ER, same basic machinery. Again, with the exception of you also in plant cells going to have a cell wall. That's a rigid structure outside of the cell membrane to give plants that, again, this is going to be composed of cellulose and other structural components that we talked about in the carbohydrates. But again, remarkably similar eukaryotic plant cell. Okay, here are our little buddies, the ribosomes, and ribosomes are responsible for making, this is the site of protein synthesis. So they are composed of two subunits, oddly enough, a small and a large, okay? And these come together, and they will actually come together around, and we'll use blue for this, a ribbon of mRNA, and I'm just going to abbreviate it like this. These are all the little bases, the nucleotides on the RNA, and this comes from the nucleus, and these things snap around the mRNA and read it like a decoder ring. Okay, so we can think of this as a decoder. Okay, it decodes the mRNA, and from the instructions on the mRNA, which come from the DNA, allow the ribosomes to make the proteins. Okay, so again, these are your protein factories, and they're on primarily the rough endoplasmic reticulum. All right, this is just showing how the Golgi and the rough endoplasmic reticulum are going to work together. Okay, so if I have protein products made from the ribosomes, they're going to pinch off in these vesicles from the rough ER, head to the Golgi, and as we said, the Golgi is going to do some final processing and packaging them up for secretion. In this case, we learned about this process here. This is exocytosis. Okay, so this is the cell making a bunch of protein products. It could be like a hormone protein or something, and then packaging it in a vesicle and squirting it out into the body's bloodstream or something for delivery around the body. Okay, so the relationship here is between what you make around the nucleus and how it gets packaged. It's kind of like the factory. You say factory, and this could be like FedEx, for lack of a better word. Okay, make it, package it, and send it out. All right. All right, another example, these are the lysosomes, okay? Lysosomes. In this case here, it's digesting this old organelle, this old worn out. Organelles wear out just like other parts of the body, and they have to be digested, okay, and broken down and recycled. In this case over here, it's the phagocytosis. Here's that phagocytosis, that's the cell eating big chunks, and you send a lysosome to this vesicle with food in it, and it digests it down, okay, into its component parts so that the cell can use the components to build something. So just two uses for lysosomes. Lysosome. Lys means to break apart, okay, and so lysosomes contain these digestive enzymes that break things apart. Alrighty. This is the cytoskeleton, really important, and it's composed of essentially three things. You've got your actin, your microtubules, and your intermediate filaments. And together these form that amazing network mesh. So if I were to draw a cell more realistically, if this is my cell, and let's say I was going to do the cytoskeleton in orange, you can actually stain this stuff and look at it under the microscope, and I mean it looks, it's like it's all over the place. I mean it literally looks like a spider web inside the cell with all these things. And remember in those videos, that Harvard animation video, you've got to have a set of highways and streets, just like here in San Francisco. If you didn't have any streets and highways, things couldn't get from one place to another, and that's exactly what's going on here. All right. Some of them are larger, like the microtubules are much larger than actin. Actin tends to be for things like grabbing and moving the outside membrane. You'll see it attached to the outside membranes and other things. The microtubules will assemble and disassemble and provide highways, large highways for things to move back and forth, etc., etc. So this is the cytoskeleton, and these are the three components of the cytoskeleton, and provides anchoring points and ways to move things around. Okay, remember these little guys? These little guys, this one's dynin, so we have dynin here, okay, and this is a motor protein that's going to be walk, and it's going to attach to this dynactin complex that then is a protein complex that will attach to this vesicle or a mitochondrion or whatever it is you want to move. And of course you have your microtubule here, and these microtubules provide a place to go from A to B, and the little dude, the dynin, walks it from one place to another. Pretty slick. Okay, so here are the different cell-to-cell connections, all right? Surface markers, okay? These are like glycolipids in the plasma membrane. When you're looking at the plasma membrane, these are things that stick out of the plasma membrane and identify the cell, kind of like a name tag. Tight junctions, okay? They surround the cell. Again, it's really just like it says. They're junctions between cells that materials pass through but not between, so you go right from cell to cell. So any place where you're going to have, for example, epithelial cells in the gut or other places of the body where cells want to send a signal directly next door, they just literally have a hole between them, okay? Anchoring a desmosome, okay, composed of intermediate filaments, okay, and cadherins. These are anchored right in the membrane, okay, and they will bind cells together, as it says, to make sure that cells in an area are not going to be floating off. Another anchoring junction, adherins, okay, they are going to be connecting cells to the extracellular matrix, okay. So this is going to be, in areas, like it says, of high mechanical stress, like the skin. So you have this connective tissue where the cells are all connected to the outside fibers so that it can stretch and bend, but it won't tear and it won't move. Communicating junction, a gap junction. You find this, as it says, in the heart muscle. Heart muscles coordinate their beating by sending signals through these gap junctions. Very similar, by the way, to these tight junctions. All right, so it's very, very similar. They are kind of like a tight junction directly between cells, but these are a specific type of communicating junction called the gap junction that is going to allow this fast communication so that the heart cells will beat in unison and in a coordinated fashion so that your heart doesn't just go quivery all over the place. And finally, another communicating junction, plasmodesmata. This is kind of like having a gap junction only in a plant. Okay, so plants kind of have their own deal, and Nothing more you need to know about that. Plasmodesmata, dealing with plants. All right, here's an example of what we're talking about. We have the cadherin. This is a protein, all right? And notice how it's anchored. And just to review our protein vocabulary, this would be, you've got this helical region here, okay? And the helical region is hydrophobic, okay? And so it's going to be suspended in the membrane. You probably have a hydrophilic head over here, and then this stuff will be hydrophilic proteins here that'll stick out. And this is just, you get two of them next to each other, and they will link together, okay, to join in this intercellular space, to join these two cells together, and of course they attach inside. This is just an example of some of those things we were talking about. And again, in plants, same idea. It's just instead of the tight gap junctions, you have the plasmodesmata. Here you have the cell walls, okay, but you provide a way through the membrane into these holes between the cell walls so that the plants can communicate. The plant cells can communicate just like animal cells do. Okay, so that is the thing. So make sure you just know what is inside the eukaryotic cell and what are their functions, okay? And you should be just fine with Chapter 4.